If you take a human red blood cell and drop it in pure water, water will rush inside the cell so fast that it will swell up and burst completely open. Human cells have no way to stop themselves from overfilling. But if you drop a plant cell into that same glass, something entirely different happens. The plant cell drinks in that exact same water, expands slightly, and safely holds its shape without tearing apart. The reason the plant cell survives is that it wraps its delicate inner membrane in a specialized protective layer called the cell wall. This outer layer is entirely missing from human cells. It acts as a highly adaptable armor that allows life to survive the intense physical pressures of the natural world. We have known about this structure for a very long time. Back in 1665, an English scientist named Robert Hooke looked at a slice of tree bark under an early microscope. He published his findings in a book called Micrographia, describing little empty rooms in the wood that he named cells. Hooke was observing the dried-out remains of long-dead cells, which looked like inert, empty boxes. However, the walls of a living plant are busy, metabolically active construction sites that are constantly being modified and reinforced. This structure performs three vital jobs at once. It provides mechanical strength to hold its shape, acts as a physical shield against the environment, and works as a molecular sieve for incoming nutrients. This shield provides the baseline stability, allowing life to move beyond the safety of calm water and into high-pressure environments that would otherwise crush or burst a naked cell. To understand how this works, we can look at the architecture of a plant. A plant builds its armor in stages, laying down three distinct layers over the course of its life. Look at this diagram. The outermost boundary is called the middle lamella. Think of this as the biological glue holding neighboring cells together. It relies on a sticky, gel-like substance called pectin to bind the cells into a single, unified tissue. Moving inward, we find the primary cell wall. This layer is built by young, actively growing cells. It uses long, flexible fibers called cellulose. Because these fibers are laid down in a somewhat random web, they can slide past one another, allowing the young cell to stretch and expand as it grows. Finally, once the cell has finished growing, it installs the secondary cell wall. This layer is much thicker, and the fibers are tightly aligned in parallel rows. The plant packs this space with a tough, waterproof material called lignin, locking the entire structure permanently into place. By combining flexible netting with this biological concrete, a tiny, pliable green sprout eventually hardens into a solid, heavy trunk, capable of supporting a massive tree. But building a thick, waterproof wall creates a logistical problem. If every cell is locked inside its own impenetrable vault, they have no way to share food or send warning signals to each other. This illustration shows the plant's clever workaround, microscopic tunnels called plasmodesmata. These physical channels punch right through the heavy armor, connecting the living insides of neighboring cells together. On the right side of the diagram, you can see how these gates allow vital supplies, like proteins and hormones, to flow freely. This open door also carries a risk, as viruses can hijack these exact same tunnels to quickly spread. The plant actively monitors these pathways to prevent infection. As you can see down here at the bottom, when a cell detects an invader, it immediately secretes a thick substance called callus to physically board up the tunnel and trap the virus. The cell wall acts as a dynamic security system, regulating the flow of information while remaining ready to isolate a cell during an attack. Plants are excellent builders, but every kingdom of life uses a completely different chemical recipe for its armor. Here we see the blueprints for bacteria. They use an exclusive material called peptidoglycan. The top diagram shows gram-positive bacteria, which use a massive, thick layer of this mesh. Below are the gram-negative bacteria, which use a thin mesh hidden between separate membranes. Fungi, on the other hand, built their outer walls using a material called chitin. This is the exact same sturdy compound found in the hard shell of a crab or a beetle. Looking at the bottom of the chart, we have archaea, microbes that often live in extreme environments. They skip the normal bacterial mesh entirely, relying on tightly organized crystal-like protein shields to survive intense heat and acidity. Because bacteria use a highly specific chemical mesh that our own human cells completely lack, we can take medications like penicillin. The antibiotic targets and destroys the bacterial wall, clearing the infection without causing any damage to our own bodies. We know the wall is strong, but that brings us back to a mechanical puzzle. If a young plant cell is trapped inside a tough, rigid shell, how does it ever get bigger? When a cell needs to expand, it releases a growth hormone that signals the cell to pump acidic particles outward, directly into the wall fabric. This sudden drop in pH activates special proteins in the wall. The acidity causes the chemical bonds holding the strong cellulose fibers together to temporarily let go, allowing the fibers to slip and slide past each other. With the structural beams suddenly unlocked and loose, the natural water pressure pushing from inside the cell forces the entire structure to stretch and inflate like a balloon. Once the cell reaches its new size, it stops pumping the acid. The wall returns to normal, permanently relocking the fibers in their new, expanded arrangement. The ultimate test of this structure comes when the environment turns hostile. During a drought, the water stored deep inside the plant begins to drain away. Look at what happens inside the cell here. As the water leaves, the inner living mass shrivels and pulls completely away from the rigid outer frame. Biologists call this physical detachment plasmolysis. Because the heavy outer wall stays perfectly intact, the plant's overall framework doesn't collapse, it simply wilts. The moment the rains return, the inner cell quickly reinflates and presses firmly against the wall, allowing the plant to stand tall again. This balance of flexibility and unyielding strength makes the cell wall the silent architect of the natural world, supporting everything from a single-celled microbe to the tallest forest on Earth.